Venga. Okay, it should be recording now. Claudia, I think everybody knows Claudia, but Dr. Claudia Cosio is it's coming to talk about the interactions between mercury and the macrophyte Elodea nuttallii. I never know how to say the words. I don't even know how to say the words that I use. Please, whenever you want, Claudia.
It's Elodea nuttallii. It's simple, and you were perfectly fine. Okay, so I decided to start this talk just showing where I live. So I'm in the University of Reims in France. It's east of Paris. So this is France in Europe, and it's east of Paris. Nobody knows where Reims is, but you all know Champagne. Okay, so that's Champagne is it's made here. It's one of the touristic attraction in the area. So this is how the area look: one yard, forest, and small cities. And Reims is also known for its cathedral, where a lot of French kings have been coronated. But it's a small city. Okay, so the story today is started more than 10 years ago, and it has been with a lot of incremental steps. So I thought it was interesting to show that you can start with a narrow question and end up looking at other things after a couple of years. So everything started in Romania. In 2009, I was invited to a field trip, and what we knew is that Babeni Reservoir, which is here on the Olt River runs in south of Romania into the Danube River. And what we knew is that this reservoir was contaminated by chloralkali plants. There is an industrial platform here, which waters goes into this small river and then discharge in the Babeni Reservoir. And we knew that there was mercury in all the compartments. So sediments, water, and fish, but no more information. And this is the industrial platform, and this is the reservoir. So we went for this field trip to study the food chain. And Andrea Garcia Bravo had made a study previously, taking care of anglers and other peoples fishing in this reservoir or other reservoir and shops. And she found that the concentration in some of those angler people were high enough to be worried about. So we knew there is mercury in this reservoir. So we went there, and these pictures I took myself, and every day, every time of the day, there are people fishing there. So eating this fish. And the goal of this field trip was to characterize the food chain. So these are the data that we got, and a few pictures showing what we collected. So on the top left, we have the bioaccumulating factor, so towards normalized to the water concentration of inorganic mercury in black and methyl mercury in gray. So water is 1, and then we have the plants, the aquatic plants, the phytoplankton that we collected with a net, zooplankton with another net, a bigger net, and different fish that we collected. And I like this picture. I use it for teaching because we really see that inorganic mercury is relatively stable in the food chain, but we see the biomagnification of methyl mercury that we expect, of course. And then the different biota that we collected, we analyzed the ratio of stable isotope of carbon and nitrogen. Here, I'm only showing nitrogen and methyl mercury. But in both cases, we identify two food chains. And one started with the plankton, which is expected. But the other one started with microphytes. And in fact, when we looked in the literature at that point, microphytes were not considered at all as an entry point for mercury, because most studies are done in the ocean, where the coastal park is really little and negligible, and nobody studied that. But in fact, in lakes, microphytes are really important biota for the ecology and for the biomass that is present. So I decided to look a little bit more in the role of microphyte in mercury cycling in this reservoir. So I collected all the green stuff that crossed my way, and here in the graph you have a summary of the data and a few pictures of what kind of microphyte we find. So microphyte means everything that does photosynthesis, and that you see by eye. So it's not microalgae, but it can be moss, it can be fern, it can be a plant, vascular plant. It's everything that is photosynthetic and big enough to be seen. So here in the graph, you have 5 species of microphytes, different ones. And the big difference that you see in mercury content is for those two are floating, so their leaves are at the water-air interface, and their concentration is very low, and these are below the water. These two that show the higher concentration, they have roots. They are anchored in the sediments. So we see that the submerged and rooted microphyte are those showing the higher bioaccumulation. And here for comparison, I draw for phytoplankton and zooplankton. So this is total mercury concentration in green, in the green bar, and the black bar here is for methyl mercury. I didn't have enough material to analyze methyl mercury in all the samples. But we see that for methyl mercury, it's very, very similar concentration in rooted microphyte and in plankton. And one species that I found everywhere in this river and that was very dense was this species, Elodea nuttallii, and it had among the highest concentration. So I thought this is interesting to study. And I did a little calculation. So I look in the literature at the density of growth of this species. And it's several thousand grams of dry weight per cubic meter of water. So if we multiply this the highest number of density that is reported in the literature with the concentration that I found in Balbeny Reservoir, we have several milligrams of mercury, including methyl mercury, that could be accumulated in this plant, in this reservoir. And of course, it's a big reservoir, several kilometers long, and with some depth. So it's a lot of mercury, in fact, a lot of mass of mercury that is taken by this plant. And of course, it doesn't stay there for two reasons. First is that there are predators that will eat the plants. And also it's a seasonal plant. So at the end of the summer, in October and November, in fact, in around 1 week, all this tissue decay and everything that is in the leaves will be released in around 1 week, according to the literature. So they can be like a peak of mercury that is released at the end of the summer. And later on, when we studied a little bit more this plant, we found that the concentration that we measure in the leaf correlates very well with total mercury in the water, but also with methyl mercury in the sediments. So it could be used also to have an integrative view of the concentration that we found in the water and in the sediment. Because of course the water, you come now and you come in 1 hour, the data is different. So it's interesting that the biota can integrate, like average what is passing by. So I started studying this species, try to understand how it behaves, how it interacts with mercury cycling in this reservoir. And one of the first things that I did was to look at microbial communities in the rhizosphere. So don't laugh. It was a long time ago. Metagenomic, metatranscriptomic were not even a dream, okay? So we took the plants to the lab and we took sediments to the lab and we grew them. And I analyzed total mercury and methyl mercury in the rhizosphere and in bulk sediment, in the pore water, and in the sediments. And we see that in the rhizosphere, it's this number, we have an increase of the methyl mercury proportion. We use TRFLP, which is based on enzyme restriction and PCR, to see the profile of the microbial community. So we extract DNA from the rhizosphere in blue or from the bulk sediment in brown, and then we digest, we amplify it with PCR, and we look at the size of the different band that we obtain. At that point, it was what we were doing for microbial community. Now, nobody does that anymore. But what we see is that the microbial community is different in the rhizosphere and in the bulk sediments. And when we look at the bands, some of the bands could correspond to some of the sulfatoreducing bacteria that we knew at the time that were involved in mercury methylation. So to know what species we had to do a 16S PCR amplification, cloning, and then sequencing, which is quite tedious, so you don't get a lot of clone. But we had a 16S gene library, and we could see that the presence of the plant increased the methylation in the pore water and also favored sulfate-reducing bacteria. So it's not only that the plant is taking the mercury that is passing by, but it's also making microenvironment in the rhizosphere that is prone to methylation of mercury. And then we wondered if this mercury will stay in the rhizosphere or could be taken by the plants. So to study this, I used a very simple system. So 2 big beakers, on one side the roots, on the other side the shoots. And then I spike it on one side or on the other side. So here is how to read the graph. It's the total mercury concentration in the shoots in green or in the roots in brown. And on top here is for inorganic mercury. And on the bottom here is for methyl mercury. When there is a minus, no spike. When there is a plus, there is a spike. So for example, here, no spike, it's my control, no spike in the shoots and the roots. So this is the background concentration that I get in the lab. Here, I spike it on the roots and I see an increase of bioaccumulation. So with inorganic mercury, an increase of bioaccumulation in the roots, but nothing happened in the shoots. Here, I spike on the shoots. I have an increase in the shoots, but also in the roots, meaning that I have a transfer from the shoots to the roots of inorganic mercury. And here I spiked on both sides, and it confirmed that inorganic mercury that is in the roots stay in the roots, but inorganic mercury that in the shoots can go to the roots. Okay, so inorganic mercury can go down. If we look at the methyl mercury, if we spike on the roots, we see an increase in the shoots. And it's confirmed, and not the other way. Okay, if we spike on the shoots, no increase in the roots, which means that in the plants, inorganic mercury can move down from shoots to roots, and methyl mercury can move up from roots to shoots, and not the other way. And we confirm this in the lab with sediments, where we grew the plants for 2 months in the lab. And when we see, what we see is that we have an increase of methyl mercury in the shoots, which confirm the both data that I showed you before, methylation of mercury in the rhizosphere and uptake from the roots to the shoots. So when we are in the field and we collect shoots, we know that inorganic mercury comes from the water, while methyl mercury comes from the water and the sediments. And because in the water we have very little concentration of methyl mercury, in fact, methyl mercury comes from the sediments. And that's why we had the correlation that I show you on one of the previous slides. So after this, we were curious to see the kinetic of bioaccumulation in the shoots. So here we expose to one concentration of inorganic mercury and to one concentration of methyl mercury over time, and we measure in the shoots the accumulation, and we do a very simple modelization, and we see that in fact we reach the equilibrium in a couple of hours, couple of days for inorganic mercury, but really 24 hours for methyl mercury. So for methyl mercury, I plotted both total mercury and methyl mercury because we have a small demethylation in the shoots. But this suggests that we have a carrier-mediated uptake, in fact. And it's something that was really not clear in the literature, and that we wondered how is mercury going inside the cells. So we did again quite a simple experimental exposure to try to determine if mercury uptake is a carrier-mediated or passive uptake mechanisms. So here in black inorganic mercury, in gray methyl mercury, and it's the graph is shown as a % of accumulation. So in fact, we have here the control, okay, which is 100%. And then we tested different treatments. We put the plants in the cold, in the fridge at 4 degrees. It slows down the metabolism. What happens? We reduce sharply inorganic mercury accumulation, but not that much methyl mercury, slightly methyl mercury. I kill the plant, I freeze dry them to be sure that they keep their geometry and their chemical composition, but they're dead, no more metabolism. Here again, very similar results as in the cold. When the metabolism is shut down, accumulation of inorganic mercury is reduced, and methyl mercury also, but less. One of the hypotheses with the metals is that they are taken up by essential metals transporter. I tested a different one, but the one that caused an inhibition was copper. If we mix mercury and copper, there is no more uptake of mercury, inorganic mercury, sorry. But methyl mercury is a different story. So we can do take the hypothesis that mercury, inorganic mercury goes in by a copper transporter, but not methyl mercury. And in fact, the transporter of copper, it takes copper as copper 1+. So I put a reducer, and here, when we do the competition with the copper 1+, definitely there is no more mercury goes into the cell. And it reduces a little bit methyl mercury, but not that much. And this is intracellular concentration, which doesn't matter a lot in the story that I'm telling you. So this shows that inorganic mercury is taken likely by copper transporter, but methyl mercury, it's somehow linked to the metabolism, but not entirely. So there might be a combination on passive and carrier-mediated transport uptake. So I also look at the responses at the molecular level, at the gene level of these plants to different concentration and form of mercury. So in fact, what we see here is concentration in the water of the spike and intracellular concentration, because we expect that to be toxic, the mercury has to go inside the cell. And usually with other species, the methyl mercury uptake is much higher than the inorganic mercury uptake. 4 times more, for example, will be expected. But for Elodea and nuttallii, it's not the case. This plant loves inorganic mercury. It bioaccumulates a lot inorganic mercury, as much as methyl mercury. So in fact, we don't see more bioaccumulation one form or the other. They are equivalent. If we spike the same amount of the water, we've got roughly the same bioaccumulation, okay? In the bottom here, you have this axis that is done here. So it's intracellular bioaccumulation and plotted against the number of genes that are significantly deregulated up if the arrow is turning on, or down regulated if the triangle has the point down. So we have the intracellular concentration and the number of genes that it affects. And what we see is that methyl mercury impacts a lot more the transcriptome than inorganic mercury. Inorganic mercury, the concentration increase, and we see no big change in the response. And methyl mercury, we have a strong response with increasing concentration, and then it's reduced. When we look at the genes that are deregulated to try to understand how they explain the toxic response, one of the things that we do is that we look at the genes that are specific to one of the condition. So this is shown in this Venn diagram. I don't know if you are familiar with these plots, but in fact the numbers represent the genes that are common to the different treatment in the middle, or on the side that are only found in one of the conditions. And what we see is that for inorganic mercury, we have little genes that are common. Most of them are found in specific condition. And for methylmercury, it's kind of similar, but we see that the number of genes increase with the concentration of exposure, which is not the case with inorganic mercury, which tell us that the toxicity targets and the toxicity pathways are different for inorganic mercury and methylmercury. And in fact, another thing that show us this study is that the genes that respond to inorganic mercury, they are almost all common with methylmercury. There is only 1% of the genes that are specific to inorganic mercury. 11% are common to inorganic and methylmercury, and 88% of the genes are specific to methylmercury. Against saying that we have different toxicity pathway so the form of the mercury determines its toxicity. So one of the I will just go fast on this one, otherwise it's very boring. So we analyze all the answer, I mean all the genes, and what we find is a very classic toxic response to metal. In fact, we see on the genes an inhibition of the production of energy by photosynthesis, which is expected for metal toxicity, an increase in sugar catabolism to compensate likely this inhibition of photosynthesis. And we see downregulation of essential metal transporter and essential metal homeostasis, likely to control and reduce the accumulation of mercury. But one of the genes that responded was in fact a copper transporter. And we remember that we had a competition of copper with inorganic mercury. So we managed to get the full sequence of the gene, and we try to functionally characterize it. So COP1 is a big family of copper transporter that we find in all the tree life, the life of tree, from microalgae to rice or corn, but also in bacteria and humans and fungi. I mean, it's very conserved, and it's a high affinity transporter that is involved in the uptake of copper from the environment. So it's this sequence, and in fact we find this different characteristic in the sequence. So we are pretty sure it's a copper transporter, but to be sure, we complement a mutant of yeast. So we use yeast, and it's a mutant that doesn't grow if you don't put copper in the media. And this is the copper transporter of yeast. So if you complement, I mean, if you transform this mutant with its own copper transporter, it starts growing even in absence of copper. Okay, it's different media, and then we add 5, 10, 20, 50, or 100 micromolar of copper, and you see that the mutant needs a lot of copper to be able to grow. And when it's complemented with the copper transporter, it grows without copper. Okay, so we use the sequence that we find from Elodea nuttallii, and we complement the mutant, and in fact, it starts growing at much lower concentration of copper, meaning that this gene is really a copper transporter. So we have a copper transporter that responds to mercury. So we look at the expression level of these genes when we spike copper or mercury. Okay, it's the same molarity. 1 nanomolar, 10 nanomolar, 100 nanomolar of copper, 1, 10, 100 nanomolar of mercury. It's a high transporter affinity, which means that at low concentration, it will be highly expressed and repressed when we increase the concentration. And what matters here is that we see exactly the same pattern for copper and inorganic mercury, which means that the plant is not able to differentiate copper or mercury. Inorganic mercury, of course. It's the same for this gene. And this is a tolerance test with mercury, but let's skip that. So we wanted to make sure that this copper transporter could take mercury inside the cells. So to do that, we use some of the models that we can use in molecular biology. Arabidopsis thaliana, it's this little plant here that is easy to transform. So we have the wild type, and we have one that overexpress these genes of Elodea nuttallii. It means that it's highly expressed everywhere, in the roots in brown, in the green, in the leaves in green. And this is the mercury bioaccumulation, total mercury. And we see that when this gene is overexpressed, the bioaccumulation of mercury increase a lot in Arabidopsis thaliana. And this one is a culture of hepatic cells, human hepatic cells, again, in white, the one that express the gene of interest, in dark, the wild type, the control. And we see, it's a fluorescent assay. We see that when we inject the mercury, we have an increase in the fluorescence, meaning that the mercury is getting inside the cells. So we know that this gene increases inorganic mercury accumulation. Sadly, we didn't find the same for the same kind of result for methyl mercury. So we still don't know how methyl mercury gets into the cells. So one of the questions that we had, maybe I will skip this. So yeah, maybe the last one. Okay, one of the questions that we had is that when we look at the distribution in the cells, in Elodea nuttallii in the lab, we find that a big proportion of the cells of the mercury get into the cell wall. Okay, so here we have the proportion into the membrane, the cytosol, and the cell wall after 24 hours, after 96 hours, in plants that are exposed to inorganic mercury or to methyl mercury. And that we see with time that this proportion increase. And in the wild, we can expect it to be even more proeminent with all the mercury bound in the cell wall. And in fact, cell wall is one of the strategies that plants have to tolerate metals. So we wondered if cell wall synthesis could be increased by mercury. So by histology, by microscopy, we simply look at the linefication of the cells after 7 days, and in fact, it's increased. Why do we care? Because in the wild, in Babeni reservoir, with the concentration that we have, we can expect all the mercury to be binding the cell wall. And in fact, it is hypothesized that if the metal is in the cell wall, it will not go into the food chain. It will be eliminated by the consumers and not being accumulated. But we know that in Babeni reservoir, we have food web transfer. So I tried to address this discrepancy by making an experiment in the lab. So what we did is that we expose Elodea nuttallii to inorganic mercury or methyl mercury, and then we wash the plants, either with Elodea cysteine to remove what is on the outside of the plant by competition and keep only in internal mercury, only cytosolic mercury. And we rinse another lot with methanol chloroform, which breaks the cells, and we only keep the cell wall. So we have intracellular mercury or cell wall mercury. And we give this to gammarids that are amphipods. It's a cousin of the shrimps, if you like, from freshwater small rivers. And we look at the accumulation after 7 days. So what we have here on the graph is mercury in the plants, mercury in the gammarids. The circles are intracellular. No, the black symbols are intracellular. The white symbol are cell wall. Okay, for methyl mercury, the circles. So if we look at the higher concentration, we see that cell wall give higher concentration in the gammarid than intracellular. And same thing for inorganic mercury. Intracellular is lower bioaccumulation than cell wall. And there were no mortality, because I always get this question of the methanol chloroform being toxic to gammarids. No, we rinse everything thoroughly, so we have no mortality. And maybe I will stop here. No, it has been long enough. What do you think, Carla?
As you like. I'm really enjoying it. So if you want to finish, go ahead.
Okay, I'll try to be quick then. So we show with this experiment that in fact mercury in the cell wall can be transferable in the food chain if the animal can eat cell walls. Okay, not every animal can eat cell walls, but gammarids can. And so we wondered if the toxicity will be similar or different for the gammarids fed with cell wall or intracellular compartment. And to do this, to analyze this, we use differential centrifugation coupled with HPLC. So in fact, we grind and then we centrifuge in several steps, faster and faster and faster. And we get a cytosol and other compartments like a cell wall, organelles, and so on. But the cytosol, we put it in the HPLC to have different fraction of proteins that we can interpret. And this procedure allows us to have 3 fractions. One that is considered metal sensitive. So the metals that it here cause toxicity to the animal. One that is biologically detoxified. So it should be tolerated if it's in this compartment. And the rest that are debris, but then we can calculate mass balance. So here we have 2 graphs. Okay, so metal sensitive is MSF. That's the one that will cause toxicity to the animal. And we have intracellular or cell wall, it's the I and the big C. And then we have control, low concentration, high concentration. That's how to read the graph. So total mercury in the 3 fractions, we see that it's increased with the concentration of exposure, and it's higher with the cell wall, which is congruent with the previous data, but we don't see real difference between the compartment. When we do a proportion of mercury, we calculate the graph here to show the same data as here, but with as a proportion of mercury in the different fraction. And here we see a difference here. For the cell wall, we have more mercury detoxified. And for the intracellular, at the higher concentration, we have more mercury that is likely toxic. Okay, so which means that we have a higher transfer from the cell wall, but that the toxicity is higher from the intracellular compartment. But to test this, we did proteomics. So we look at the different proteins that are more or less abundant with the different treatments. So here we have intracellular low concentration, cell wall low concentration, intracellular high concentration, and cell wall high concentration. And again, the Venn diagram. And we see that nothing is in common. In fact, most of the proteins that are affected are in the different treatment, and especially intracellular compartment high concentration. So the one that is supposed to be more toxic has more effect in the protein. So it's likely more toxic. And here it's another way of showing that the responses are specific to each treatment. Okay, red means higher, more abundant protein. Green means less abundant protein, but we see that the pattern is different. Each compartment, each concentration is totally distinct. And these are the functions that are attributed to these proteins, but I will stop here. So to summarize, we started studying Elodea nuttallii more than 10 years ago. And what we have learned on the way is that it changed microbial community. It increased methyl mercury in the rhizosphere. It transferred methyl mercury from the sediments to the shoots and to the consumer. It accumulates inorganic mercury and methyl mercury very rapidly. Inorganic mercury is taken out by a carrier-mediated process, and Elodea nuttallii is involved in the trophic transfer of methyl mercury to the food web. So it represents a risk of increased mercury methylation and remobilization from sediments and subsequent transfer into the food chain. So all this was made with a lot of different students and collaborators that a few of them are here in picture. And well, thanks to them and thank you for listening.
Hi.
I could talk for hours on this topic. I'm sorry.
Sorry?
I could talk for hours on this.
No, that's good.
It was a little bit long, sorry.
It really seems like you know your stuff after 10 years of studying the same plant.
Yeah, it's like next.
I have some questions,
Sure.
like in general. So you said that you studied the same plant, but is it the same plant from the same place? Like you collected it once and now you replicate it at the lab, or you go
Yeah,
a couple of
yes.
years or every season, or?
In fact, I collected here. This is Lake Geneva, and it was in front of the institute where I met Andrea, Forel Institute, and there were a small harbor full of Elodea nuttallii. So I didn't collect the one that was in Romania, because it was too much driving. It will die in the process. So I collect it here, and then how you say that? You cut it and you plant it and it grow again.
Yeah, replanted.
Yeah, it's clonal reproduction.
Yeah.
So in fact is it's this strain that was collected in front of Floral Institute that I kept in the lab, because if you work with the wild plants, in fact there are only a few months where they're growing. In the winter you don't find them.
Yeah. And just so the specific transporter for copper, was that found already in these plants, in the ones that you collected in Geneva?
You mean the gene?
Yeah.
No, in fact, when I start working in this plant, they I don't remember, they were maybe 12 sequencing in NCBI, and all were Rubisco. So no, the sequence we didn't have, and in fact, well, you know that with Illumina sequencing you get chunks.
Yeah.
And so we had to do inverse PCR to get the full sequence. It was tricky to get it. So no, it's the one of the sequence that I put in CBI that we find. So no, we didn't have the full sequence, and we were not sure that it was a copper transporter, because by homology,
Yeah,
you're never sure of the function. So that's why we did so many experiments to be sure that it's a copper transporter first, and then to show that it really is able to take mercury in different species, in different kind of biological assays.
Yeah. So you found, so it's, do you know if it's specific to plants, that copper transport, or if bacteria have it as well? No, right? That can be like it has to be plants.
It's a family that you find in all the tree of life.
Huh.
So you will have homologs everywhere. I mean, in every living organism, because copper was really high at the time of life beginning on our planet. So in fact, it's likely heritage from the very beginning of the life on Earth, and copper is essential to many proteins. So it's very something that is common to all kind of living organism. So I know there are homologs in bacteria, there are homologs in mammals, all kind of plants, fungi, everywhere.
Because I've seen people that work, for example, with like arsenic and mercury, or selenium and mercury, but I don't think that I've seen like bacteria. I'm talking about bacteria because that's what I work with, but like co-assays to see copper and mercury at the same time in culture. That's interesting. I'm monopolizing.
Yeah, but I think they were studies on this, but all the studies, and it's inorganic mercury. It's not methyl mercury.
Yeah.
Okay, so now we are more interested in understanding in bacteria how methyl mercury is made, how mercury is taken in and out from the cells, and so on.
Yeah.
I know that in microalgae, it's not copper, it's more zinc that does competition with inorganic mercury uptake. So it's also species dependent, depending on the metabolism of the species and the metal homeostasis. It will be more copper or more zinc or and so
on. Carl has posted a comment on the chat if you want to read it directly.
Yeah. Yeah, that's a good point. In fact, the copper transporter is so COP1 is coupled with a reductase. So at the surface of the membrane, you have the reductase that takes copper2, turn it to copper1, and give it to COP1 to go inside the cell. And inside the cell, it's oxidized again to copper2. And copper2 then is useful for many kind of proteins to do oxidoreduction. For mercury, we wondered if it will be the same. And in fact, I didn't find how to test it, I have to say. Yeah, it's a good point. If it works the same way, it should be mercury 1 reduction to mercury 1 and goes inside. But how to show it? And we have trouble to publish this work because they're asking for a lot of controls. And one of the experiments is patch clamped, which is very, very tricky and difficult to do a lot of experiments. So it's taking ages to answer to reviewer comments. So it's not published yet, and yeah, we are struggling to get it published. So if you have an idea, Carl, how to test it, I will be happy to try.
I have a question. I mean, I have so many questions, but if anyone else want to talk still about the same question that Carl had, go ahead. If not, okay, awesome. One thing I wanted to know is that, first of all, awesome talk. I mean, I'm so excited. I'm glad I came because I learned a lot. One was like, have you ever able to like knock down this gene and then see that what will happen to like the plant and if still uptaking, what, you know, or like not, I mean, or like overexpress it, or take this gene to like a simpler plant or something and then see if they act differently or no?
Elodea nuttallii, it's impossible to do molecular biology with it. It's an aquatic plant, and you know, when you do transformation, then you have to grow them in axenic condition, and it's impossible. With Elodea nuttallii, it's impossible. It always gets contaminated. So what we did is that we use Arabidopsis thaliana.
Yeah.
And in fact, we have the knockout mutant for the homologous gene in Arabidopsis thaliana, and it accumulates less mercury, but it doesn't grow very well because it suffers from copper deficiency, and copper is essential. So it's tricky to play with the knockout mutants of copper transporter, because the plants are not very happy.
Yeah.
So,
can you just like give them like an available, yeah. I want to say, can you just like give them some available copper, like a spray or something that they still have their copper, but you know, they won't need to think about copper because it's bioavailable for them, and then you do whatever you want with your gene and mercury.
But you still need to put it like inside the cell, and that's what this gene does, no?
Yes, it's the high affinity transporter. It's really like sensing what is in the media, and then if it's low, the copper is low, it will be highly expressed, and if the copper is high, it's repressed, and it takes into the cell.
The other way was like overexpressing it. Like if you knock out killing it, you overexpress and see if it do it more, you know, if like, or like add more copies.
Yeah, but
I mean, it's just like easy in theory. So
no, but that's what we did. In fact, let's see. So this graph here is about that. In fact, it's an overexpression. 35S is a cauliflower mosaic virus promoter, and it's very efficient at expressing the genes everywhere.
Yeah.
And in fact, in wild type, the COP1 is only expressed in the roots. Okay, the normal pattern of expression for COP1 is in the roots and not in the shoots. And it's regulated by the environmental concentration of copper. So here you see the wild type, and here you see the overexpressing mutant. So
that's a very good result. Yeah,
so
sorry, I missed this, but like it's very
too many
awesome.
results.
Yeah, this is awesome. Like, yeah, I mean, like you actually see this. It is huge, I guess. Like, I don't know. I love this.
Yeah, me too, but it's not published yet because reviewers ask us thousands of controls.
But that's awesome. That's very good. Yeah.
Yeah, I wish I will have this for methyl mercury. This will be really cool. But yeah, for methyl mercury, we are still trying to understand how it works.
That was my other question. Is like, can the plant itself have things that, you know, to convert inorganic to methyl mercury, or no? Like it's just it's because that would be crazy big.
Yeah, but no, it doesn't look like it. There is one paper from a Swedish team where they show mercury methylation in I don't remember the name of the species, but it's a water spinach, some of the Asian vegetables that are eaten in Asia. But it looks more like an artifact of their experimental process. In most plant and algae, you see demethylation. It's
actually there's multiple studies that use plants to bioremediate mercury.
Yeah, it's more demethylation. The only exception is in the rhizosphere, but this is microbial methylation. But because the plants take the oxygen in the rhizosphere, it makes a suboxic microenvironment that is really nice for microbes that do the methylation. So in fact, you have mercury methylation in the rhizosphere. It has been shown by us, but also in rice and also in this Amazonian floating microphyte where you have methylation in the roots, and it's more like the roots microenvironment than the plants itself.
That's interesting. I just sent a paper to everyone if anyone wants to know about passive and active uptake of mercury by microbes. That's actually a good paper. I don't understand it, but like you read it. I have also other question about like the plant itself. Like when you start like that, your first few slides, you're talking about like, you know, you saw this Edola. How you spell it? Edolia? That was high.
Elodea, yeah.
But did you also try like overall, like see the chlorophyll like A as a proxy to, you know,
I'm just trying
oh, okay.
to get my, it's like frozen. I can't, can I get out of the presentation? I don't know how to do that. Ah, voila. So if the chlorophyll content was, that's your question?
Yeah, I just wanted to know, like, if you check the chlorophyll A, and also like, because in my system, I'm seeing that whenever methyl mercury is high, my chlorophyll A is high. So I just like wanted to know if you see the same thing, or does it make sense, or is my model correct?
We didn't look at that at the beginning,
Okay.
but I have some data. I think, no, I didn't put it here. What we used is to as a proxy of the stress level, we use photosynthesis activity, chlorophyll content, anthocyanin, that is an antioxidant, and lipid peroxidation, the activity of antioxidant enzyme, things like that. We have used that are really classical in ecotoxicology to look at the stress level. And in fact, we often have a bell-shaped answer. Like when we increase the concentration, like here is for the genes, but for the chlorophyll content and the other endpoints that we can look, we also have this kind of shape. Like it increase with increasing concentration, and then we pass a threshold or something, and then it goes down. And it's really time dependent also. If you do it at 2 hour, 24 hour, or 44 hour, it's different. It's acting a lot, but it makes sense to look at the chlorophyll and the photosynthesis, because in the chlorophyll you have magnesium, and the magnesium is replaced by the metals. That's a very classical toxicity target of metals. So
interesting.
So you have a hem in the chlorophyll with a metallic atom, but it's weakly bound for chemical reason. I'm not a chemist, so I will not go too much into that. But the metals like cadmium, mercury, zinc, they replace it, and then the chlorophyll is not functional anymore, which affect the photosynthesis.
At that threshold, yeah. But for now, like, yeah, chlorophyll A goes up, mercury might go up or not, and then the moment its threshold hit, it just like you will see a drop.
Yeah.
That's so interesting. I have some other question, but I will allow other people to ask theirs first. I will come back if we have time.
Yes, please. If anyone has any more questions, just jump in. I have
hi,
sorry, you go ahead.
Yeah. Okay, sorry. Claudia, I have one question. I think you found that a gene that's NCOP-T1 could be a transporter for organic mercury, but you didn't find any transporter for methyl mercury. Can you make more comments about that? For what reason do you think you didn't find any methyl mercury transporter, or what potential transport mechanism you think that methyl mercury would be?
What we have found, okay, so we did RNA-seq transcriptomic, and we find this gene for copper, and we already had data showing that copper was a good competitor to mercury. So we had an array of evidence pointing to this suspect. So that's why we chose it. But in fact, in the competition, if I come back here, for methyl mercury, nothing really reduced the uptake. There is some metabolic uptake, slightly, maybe 40% maximum is reduced when the plant is dead. So there is a passive uptake, clearly. Okay, because we have bioaccumulation when the plant is dead, with no metabolism here. And for the rest, for the one that is linked to the metabolism, what I think, but I have no proof of it, is that it's related to amino acid uptake. I think, based on the literature, that methyl mercury bind to cysteine or to thiols.
Yes.
And I will hypothesize that it's taken into the cell, bind to cysteine to thiols. But it's not as easy to prove it, because amino acid uptake is not so well studied. So we don't, copper is very well studied. We know perfectly how the homeostasis of copper works. So I didn't go into detail, but we have here the transport pathway of copper in plant cell. We know all the actors. So for copper, it was quite easy. And I could find people that have the mutants, and the databases are very full of sequence, so you can check that you have the right sequence, and so on and so on. So copper was relatively easy. Amino acids uptake is like a dessert. you don't have so much information. So I have several amino acids that transporters that are identified in the transcriptome, but we are not advanced.
And also, so through thiols and cysteines, you can also find mercury without methyl, like just mercury. So it can also be uptaken in organic as well.
Yeah.
So that shouldn't take the balance, no?
But I think it is a matter of affinity. You know, it's competition. If the mercury transporter is highly expressed, I think the affinity for inorganic mercury will be so high that it might take everything. And if the transporter is lowly expressed, it can go through other transporter and pathways.
Yeah.
But yeah, sure. Thiols will bind inorganic mercury as well, for sure. But based on the current knowledge, I will look at the amino acid transporters. But it's not as easy, because it's not studied as well.
Yeah. Thank you very much for your answer. That's great.
We can extend like 5 more minutes, but at 5:30, we should probably let Claudia go home if she wants to. So if you want to ask your last question, Sora, that's fine.
Yeah, that my last question was like a simple one. I was just like wondering if, because I think in North America we call your plant like an invasive plant, but like, is it invasive in France, or like how is it?
It is actually, it's from North America.
Is it?
So yeah, it's originated from North America, and it's invasive worldwide in temperate fresh waters. So
You're welcome.
Okay.
yeah,
Carl says you're welcome.
and in fact, there are two species. There is Elodea canadensis and Elodea nuttallii, and both are in Europe. Canadensis has been there for more than a century, so it's considered acclimated. And Elodea nuttallii, I don't remember. I think it came in the 80s, so 40 years ago or something like that. And yeah, it's spreading days after days.
And I also have a small question. At the beginning of the presentation, you said that because it's a seasonal plant, there is a point in the late autumn that it loses its leaves.
Yeah, it degrades.
Yeah. So when you went and measured concentrations in like fish and stuff, did you see if you found that high levels of mercury when the plant it's degraded in the water, or
we never went in fall. We always went in summer, so when it was full of plants. But I think it's something that should be looked at some time, because in the literature it's written that it degrades in 1 week. It's gone. So imagine you have several milligrams per cubic meter of water, thousands of cubic meters of water, and suddenly it's like released in 1 week.
Amount of mercury released in a very small time.
Yeah. So it could do like a acute exposure of the biota that is there.
Yeah.
Just because it's released.
Exactly. Yeah, that's what I was thinking. If you ever went to measure specifically that.
No, we didn't. Romania is pretty far.
Yeah, I know. That's why. That's exactly what I say. Like, were you there like multiple times, or was just like a
we went, I don't know, 4 or 5 times. I don't remember now. I should look, but always in summer.
Okay.
When the plants are growing, in fact, we were looking for the food chain, and the plants, so we always went for summer.
Okay, I see. Well, it's 5:30, so I think we should leave it here. But if anyone has more questions, sure, Claudia will answer you by email or something.
Yeah, of course. Don't be shy.
So thank you. I'm gonna stop